Our speaker today is Dr. Drew Kitchen. He is an assistant professor at the Department of Anthropology here at the University of Iowa, and the topic for today's meeting is Archaic Ancestry. And so Drew's primary research focus is understanding human population demographics and migration patterns, and also has an interest in understanding evolutionary history of infectious diseases. And so he uses computational models to understand these principles, and I think is going to talk a little bit about one of 23andMe's features is showing percent or number of Neanderthal variants, so I think he'll be discussing some of that today and some other topics related to archaic ancestry. So please join me in welcoming Dr. Drew Kitchen.
Thank you. You can hear me loud and clear, which is great, because usually I'm a soft speaker. Thanks for the introduction. This is a great opportunity. You guys probably don't know me, but I see some familiar faces that I've seen around the community. So it's great to be here. I'm going to be talking about archaic ancestry, which is just the latest in the series of talks in this DNA interest group. I envision this as being a kind of a semi-formal kind of presentation, where if you guys have questions, you could ask me to explain something a bit more. I guess definitely afterwards we're going to have questions, and there's a microphone that needs to be circulated. If you want to ask questions during the talk, I can repeat your question so that we get it on tape. I'm a computational geneticist, so I don't have a wet lab. My lab is like 14 big computers all linked together, and I crunch the numbers behind the scenes. And I have a tendency to get really excited about kind of statistics and computational stuff that might be a little obtuse, so if you have a question, please let me know. All right. Now, I'm an anthropologist, and we're interested in where people come from, who they are, how they identify themselves, and that's what many of you are interested in. And so the first thing we have to think about when we think about archaic ancestry is, who is archaic? How do we define what's archaic? So this is a family genealogy of human relatives after we split off from chimpanzees. If we had chimpanzee here, it would be coming way off here and being a very distant relative. And these are all fossil species of hominins. Starting right about here, we think that these guys are definitely bipedal, and these are all hypothetical relationships. All right, so these would all classify as archaic. Now, we're not really interested in most of these when we talk about DNA ancestry, for the simple fact that they've been extinct for millions of years, and we can't get DNA from them. All right, and we'd love to get DNA because you see these little dotted lines here, and there's question marks here that would resolve these issues, but we can't. So the things that we're mostly interested in when we talk about archaic humans are those things that are most closely related to us. So this is us, and we're mostly talking about these guys, all right? So archaic in the kind of genetic ancestry world really deals with Neanderthals, a group that's not up here called the Denisovans, and most recently we're getting some data from Homo heidelbergensis, which we think is the ancestor to Neanderthals, all right? So when we say archaic, it's code for these guys, okay? So in this talk, I'll be interchangeably talking about archaic and Neanderthals, and that's generally how it's used looking at 23andMe. Now, at the end, we're going to talk about some other human populations that no longer exist as coherent human populations, and that kind of, I should say, approaches what we think of when we think in archaic. All right, so Neanderthals are really what we're talking about. Now, why are we really interested in Neanderthals? First of all, you'll see in other slides that they're just fascinating. You know, in and of themselves, they're a pretty interesting human population. But this is another diagram of human species and where they are through time and then geography. You'll notice that archaeological evidence suggests that modern humans and Neanderthals might have overlapped in time and space, all right? So why is that important from, look, if we're interested in DNA and genetics and ancestry, it's the potential that they might have interbred, all right? They're living in the same place at the same time, possibly they might have had contact, it means they might have interbred. Now, it doesn't make any sense if you have something that went extinct long before modern humans, all right? You could go look for that, and you might find a signal, and that would completely rewrite all of our archaeological history. It's much more safe to think about those things that we actually have physical evidence that are in the same place, the same time, or similar periods. All right, so that's really why we're pretty much interested. So we're interested in these archaic humans that lived within the last 200,000 years. Okay? So archaics are extinct human populations that, for our reasons and self-interest, overlapped in time and space, possibly with modern humans after modern humans moved out of Africa. All right? So that's really what we're talking about. So that's what archaic is code for. Now, archaic humans populations show up basically in the middle of the 19th century. All right? Before that, hadn't really thought about it. I mean, there have been long thoughts about where humans came from and whatnot, but not in the sense of an organism from which we evolved, right? So this is one of Darwin's big ideas, was that modification with descent, natural evolution of species, and about the same time we start finding archaic humans. Okay, so if we weren't thinking about some different kind of human that modern humans evolved from until that time. Okay, and so this is a pretty interesting point. It's wonderful, in kind of the end point of Enlightenment thinking about where we came from and where we started out. And the initial thoughts were these archaic species are just dead ends. Like, they died out. They were probably outcompeted by modern humans. We're better than them. It's kind of, some people still thought of lineal evolution, of that's the replacement of better and better and better, in this case, more thoughtful, more cultured populations. And in fact, the earliest reconstructions of what Neanderthals look like were of kind of decrepit individuals. This is partially due to this overriding sense of, populations are getting better and better, and we're moving up toward this kind of European sort of elite cultural pinnacle of humanity sort of thing, and that the populations that were replaced were these decrepit kind of weak intellectual individuals. It was also a problem with the archaeological record, is that one of the first finds we found was an old man who had osteoporosis, and it turned out that when you reconstructed, he was kind of hunched over and didn't have any teeth, and so this is one of the issues of when you find scant evidence, you might, what happens if the last fossils of modern humans happen to be, the Chicago Bears linemen, right? Everybody think we're just enormous guys that are huge and everyone's super tall, right? But that's not the case. And so this kind of played into our earliest interest in our archaic humans, okay? Now, since then, we found many, many more individuals. We have a couple of hundred, I believe, Neanderthal individuals that we've identified. So we're not talking thousands, we're talking, between 100 and 200. And our view of them has dramatically changed. So archaic human populations, Neanderthals, probably Denisovans, though we have incredibly little information about Denisovans, they spread over a large range of Europe and Asia into the Middle East, occupied these areas for thousands of years, successfully dealt with incredibly hostile climates, and actually showed cultural adaptation over time. Their culture changed over time. They became more, they had developed new technologies. Toward the end, they started developing technologies that were very, very similar to what modern humans at the time were developing. All right, so our view now is that these archaic populations were very much more like us. All right, and so throughout this talk, I'm going to try to present to you this idea that they're more like us than they aren't like us, and that the fact that we share ancestry suggests that, all right, that they're not some other thing, but that they are very similar to modern humans. So this is what we're talking about, all right? So once you move from here to here, just intellectually, the possibility that modern humans interbred with Neanderthals increases dramatically, right? One thing that people don't think about much, and I think and it's talked about much less, is that there is massive amounts of mate choice in humans, and at some point, modern humans chose to mate with Neanderthals, and vice versa. So they saw each other as behaviorally possible mates. All right, that's unlikely to happen if you have a decrepit, weak-willed population, right? And so this tells us something. And people don't talk about this too much, but in anthropology we do. We don't talk about it much outside of anthropology, but our conferences we do. So this is a major shift in how we think, right? And this is what we're talking about today is a final step in that. Now, before we had full-on genomic technology, and we were able to get genomes from ancient humans that lived thousands of years ago to tens of thousands of years ago, we now have mitochondrial genomes from populations that live more than 300,000 years ago, possibly 500,000 years ago. There was initial evidence that humans and Neanderthals didn't interbreed, all right? So the first evidence comes from mitochondrial DNA. So mitochondrial DNA is inherited maternally, right, from the egg, so through the egg of the mother, and the male sperm does not contribute any mitochondria. And so if you trace mitochondrial relationships back, you're actually tracing maternal inheritance. Okay, so it's one marker. So I have my mom's mitochondrial genome, and I will not pass it on to my child, all right? Barring some technological feat, which has now become possible and is approved in the United Kingdom, where you can have 3 people contribute to 1 offspring by mitochondrial genome transfer. That never would have happened, right? So we don't have to worry about that happening in the past. But what we did is we were able to get mitochondrial genomes from modern humans. So the Africans have the longest branches. They're the closest to the root. So they show the most diversity. And then we have some ancient Neanderthals here. And you'll notice that the lineage leading to Neanderthals is distinct from the lineage with humans, modern humans, all right, which shows that there was this is suggestive that there's no interbreeding between modern humans and Neanderthals, all right? This was preliminary data over a decade ago, all right? And this suggested that, hey, this probably didn't happen. The problem is that it's only one locus, all right? So your genome is composed of 3 billion bases, right? And you have 23 chromosomes. And when you produce gametes, there's a recombination occurs, right? So each chromosome that you produce in your gamete is a mixture of the 2 copies of each chromosome that you get from your mom and dad, all right? And so each one of those is an inheritance unit, essentially. Now, in your genome, you have millions of these. And so they can all follow different inheritance patterns, all right? So I might get one section of my genome from great-great-grandpa Kitchen, and then the other side of it would be from great-great-great-grandma Albert, my mom's side of the family. And then when I pass off to my offspring, I would only have one from Grandma Albert in that section and not from Grandpa. But the next section could be from Grandpa Kitchen. All right, so they have distinct evolutionary histories, each one of these parts. These units get split up, and so each has their own genealogy through your family. So it's actually very likely that if you go back more than 8 or 10 generations, you have ancestors that have no contribution to your genome. They just haven't. They're not found in any of your genealogies. Now, what's important is that mitochondrially, it's always from your mom. This is one locus that you always get from your mom. And instead of the millions of loci that you get, like if you go to 23andMe, now they're sequencing over a million different little mutations in your genome. This is effectively just one. So it's a very small fragment of the story. It's like taking one page out of the book, but it was as good as we could do at the time. And so this was initially touted as evidence that this page in this book of our genome said that Neanderthals and modern humans were entirely different lineages. So they separated and then never interbred. Right? And in fact, when you look at, we've since fleshed this out. So you still have Africans that are the most diverse, they're closest to our common ancestor here. You have Asians and Europeans because we came out of Africa. You'll see Neanderthals are still over here, even though we've fleshed this out. We've gone from 3 to maybe half a dozen. We actually have some Denisovans now. Denisovans are the species of human, archaic human, possibly closely related to Neanderthals, for which we originally had a tooth and a finger bone. That's all we had. And then we have these populations of archaic humans that seem to have been deposited in a cave in Spain, in Iberia, where they threw a bunch of bodies into this cave over 300 to 500,000 years ago. We've gotten DNA from them, but it's all mitochondrial DNA. And this suggests that there's no interbreeding between Neanderthals and modern humans, or Denisovans and Neanderthals, or these SEMA individuals. All right? So this is what the initial thought was. All right? So this is consistent with Neanderthals. It might be behaviorally more modern, but there's no interbreeding, no offspring are produced from this, no matter how modern they might be. Maybe it was there's a reproductive barrier genetically. Maybe the overlap in time was very short and there just wasn't enough time for a significant number of breedings to occur. You can imagine people having offspring and their offspring dying over a period of years just by chance alone. Right? So one thing is if we're going to see evidence of interbreeding, we're going to have to see a lot of it. There's going to have to be a lot of interbreeding occurring. Or maybe they're behaviorally distinct. They didn't recognize each other as mates. There's all these reasons. But this is consistent, right, with our a priori expectation at the time that Neanderthals and modern humans were just different. Right? And then that changed. And that's when we started genomics. All right? Genomics is the study of all the genetic material, not just the mitochondria, not just the Y chromosome, but all your autosomes, the 22 chromosomes from which you inherit one copy from each of your parents. All right? And where there's one genealogy that relates everyone with mitochondrial DNA to each other, and there's one genealogy that relates everyone with Y chromosome to each other, you have multiple genealogies in your autosomes and the main 22 chromosomes. So each one of those genealogies is an instant of evolution. All right? So instead of just looking at one page in a book, now we're looking at all the pages in the book together. And this didn't really start until roughly about 2010. Green et al. Has a paper put out by Svante Pablo's lab where they sequenced 3 individuals from Vindige. It's about 40,000 years old. And they were able to look at the first draft sequence of the Neanderthal genome. All right, so this is when archaic genomics, human genomics, really came of age. All right, now, since then, shortly thereafter, they published a genome of what we call a Denisovan. All right, this was, there's a tooth, was originally used to get mitochondrial DNA. There's a finger bone that is roughly the size of a dime or so from a cave in Siberia called Denisova Cave. And they sequenced the genome from this, and actually the genome from this is almost on par with most modern genomes that we would have gotten at the time. It was an incredible technological feat, better than the Neanderthal genome. And it turned out that this was an entirely new archaic human population that we'd never seen before. And there was no archaeological evidence for it. And so this was kind of astounding. Now we're discovering new human species, possibly species, because if Neanderthals are a species, Denisovans are a species. They're that distinct. But for the first time in the anthropological record, anthropologists decided to not name a new species. All right? Usually the way you get into a big name journal is you name a new species and then they always have to cite you and you get into Nature and Science. These guys, Svante Pabo said, we're going to call these Denisovans. Just like we call people from Europe Europeans and people from Asia Asians, we're calling them Denisovans. We're going to know them geographically. We're not going to say they're a new species because we know from this paper that Neanderthals did interbreed with modern humans because we found parts of this genome from these Neanderthals sprinkled throughout Eurasians. And so maybe this whole idea that there's this separate population, that there were barriers to reproductive, like what we see when we define species through the biological species concept, where these two populations cannot possibly interbreed, all right, to produce fertile offspring. Maybe we should just talk about these as archaic humans and just kind of skirt the issue. They're all humans because they might have provided DNA. And in fact, we did find in this genome, and I say we collectively as a field, not me personally, we did find that there were some populations, especially Melanesians and East Asians, that have Denisovan sequences in their genome. Right? So this is really where archaic human genomics comes from. And nowadays, everyone's doing ancient DNA. I'm working on papers looking at Native Americans 8,000 to 9,000 years ago, up in Newfoundland and things like that. And it's really, really sparked off since then. And what it's told us is that our ancestry and our history is really, really complex, much more than we like to think of these little discrete little units. All right? So this is just a long-winded way of defining archaic and why we're actually interested in what's archaic and where we've come and where we are now. All right? So the question that we're all here to consider is, how do we know what proportion of Neanderthal are you? All right? So we've set the stage, we've defined what archaic is, and we've defined why we might be interested in it, and technologically where we are, and we know that some of us carry around some archaic DNA in our genomes. But how do we find this out? How do we go from ordering this kit, hopefully you got it before the price jumped by about $100 when they get their FDA approval again. How do you go from ordering this kit to them taking your saliva and putting on DNA shrinking and putting on this kit, this chip, then finding out all of these little mutations that occur throughout your genome. So this is just a map of your genome, and it's a heat density map of where all these mutations are occurred, where it's dark blue, there aren't very many of them, where it's yellow, there's a lot of them. So you can see that this is what the data look like, and there are large portions of your genome that they can't see at all. All right, they don't tell you that. Like we actually haven't sequenced the entire human genome yet. There are parts of it that we actually can't get to. All right, that we don't know how to characterize. All right, into this, which is me. This is me in 23andMe. All right, so you can see here that they've mapped out that I'm probably what everyone would have guessed, is that I'm 99.3% European, and I'm basically from England, from Europe, from Britain. All right, so this is what we're going to be talking about today. And how do they go from getting this information and then making an inference about what percentage Neanderthal you are? All right. Well, what percentage Neanderthal you are takes us to a concept called incomplete lineage sorting. All right. And this is where I need you guys to ask questions if you aren't following. Okay. This is a complex concept for anybody, but I'm giving it a try because I believe in us. All right. So we'll take an example which I borrowed from a blog. So if you guys are interested, it's a blog dealing with biology and coming with concepts. And this is a linguistic analogy. So consider that back before the colonies, the North American colonies were settled, there was British English, some ancestral form of British English, right? No language doesn't change, it's never set in stone. Well, okay, some languages are set in stone, but this is a spoken language, okay? And at some point in time, you have alternative uses for words, and in this case, alternative spellings, right? The famous color sort of spelling. You can think of center, right, and different spellings between British English and American English. At some point, you could use both, right? Having gone through a bunch of documents, historical documents from the origins of the colonies and charters and whatnot, this is definitely true. You could use both. Now, at some point in time, these language communities start breaking up. So first you get English colonists leaving England and moving to the New World, populating the New World, and then you start getting separation between the colonies. And at some point, the use, in each one of these populations, the use of one of these rules changes, is lost. All right? And then the other rule becomes fixed. And this is usually, for the purposes of this example, we're going to assume that this is random. All right, so it's just completely random whether it happens. You flip a coin, and one day everyone decides, okay, we're going to spell everything with O-R. And then another population flips in and says, okay, we're getting rid of O-R, and we're going to use O-U-R. Now, what you're left with is a tree that looks like this. So O-U-R spellings persist in Canadian English, persist in British English, and die out in American English. O-R spellings persist for a certain period of time in British English, but die out. They persist all the way to the present American English, and now are the only option in American English, and die out in Canadian English. Now, if we used just these rules of what populations use OR, what use OUR, as a marker to say who's more related to who, we would say the Canadian English population is more closely related to British English population, despite the fact that we know historically that they separated from each other after they had separated from Britain. This isn't a perfect analogy, it's a little bit more complex historically in this, but this is This is essentially what incomplete lineage sorting is that you have a known relationship, and you might have genes whose genealogies don't follow that, all right? Because there's ancestral diversity that's being inherited, okay? And then at in each population, one of those ancestral alleles becomes dominant and fixed, and one is lost. Now, if we switch this over, and now we start talking about, question?
Yeah, so just, I guess, links back to biology. This is kind of similar to, say, two phenotypes, two similar phenotypes showing up in two completely disconnected organisms?
So the question is this similar to two different phenotypes showing up in different organisms, but showing up independently of each other, right? That's more convergent. So that's usually what happens when you have the same environment in two different evolutionary trajectories. So you have two different lineages, and they develop the same answer to living in a certain niche. So this is different, but it's a good question. So here we have this ancestral population, and there's a big A variant and a little a variant, and it's inherited by all these descendant populations. Now what happens is that over time, one of these variants will drift out in each one of these populations, okay? And then you'll have the same relationship of this marker of relatedness between populations as we did with the language marker, right? So if we reconstruct this here, we'd say species 3 is most closely related to species 1, right? Because species 2 lost the big A, whereas species 2 has only the little a, whereas species 3 has lost it, and so has species 1. And so to get the relationships between species 2 and 1 and 3, you have to go all the way back here, and then come back down to here. And that's the relationship between species 2 and species 1. So it looks really, really old, right? But in reality, the population split very recently. Now, we expect this to occur randomly. So 50% of the time, here we showed species 2 being distantly related to species 1 and 3, right? Having to go all the way back here. Well, this is a random process. So given 2 of these alleles, if we did this again, we'd expect 50% of the time that species 3 would be closely related to species 2 and distance from species 1, all right? And because it's a random process, 50% of the time, it would favor 3 and 2, and 50% of the time, it would be 3 and 1. Now, why is this important to looking at Neanderthal DNA? Well, it's because Neanderthals are species 3, and modern human populations like Africa are here, African populations, and Europeans are here. And if Neanderthals never introgressed, never brought some of their alleles via mating into one of these populations, then the number of times species 3 is related to species 2 versus the number of times species 3 is related to species 1 should be the same. It should be completely random. It should be 50-50, all right? And this is essentially incomplete lineage sorting, okay? So how do we go about figuring this out? Well, we find positions in the human genome where Neanderthals differ from chimpanzees and where one human population differs from Neanderthal. So you have a situation where it's either A or C. It's essentially one of these single nucleotide polymorphisms, one mutation that can either take one nucleotide or a different nucleotide, one allele or the other. Right? You make sure that chimpanzees have the ancestral state, Neanderthals have a different state, and then in modern human populations, it's also variable. It's either one or the other. So it's little a or big A. All right? Now, what we're interested in then is that once we have these, we count how many times one human population matches with the Neanderthal population versus how the other human population matches with the Neanderthal population. I know this is conceptual, but I have a plot. And trust me, this is everything you need to know. All right? I say this jokingly. This is, so we have an ancestral population of humans and chimpanzees. So here are the humans, here are the chimpanzees. Okay? And so we have one allele A. And then we have a mutation that occurs, and now we have our 2 allele set, A and B. Chimpanzees have A. This mutation can occur anywhere. We'll just say it occurs here. So it's possible for all the humans, including Neanderthals, to inherit the B, this new allele that occurs. So now we have A and B in both these populations. Neanderthals split off, all right? A allele is lost by chance. Right, this is the coin flip. It could be A or B. One of them is going to get fixed. Random genetic drift occurs, which is what we call the random process in which allele frequencies just randomly shift over time. Imagine a couple all have all daughters, then they lose the Y chromosome. Just completely random whether you have a daughter or a son, completely random. If someone has all sons, that the mother's mitochondrial DNA lineage is gone from this earth, all right? And so that's just randomness, okay? So this occurs everywhere in your genome. And so in this case, the A is lost. So now we have the B allele. We still have these two alleles they're sorting through. In modern human population 2, the B allele is lost and A allele is found. A allele persists. In this one, the A allele is lost and the B allele exists. All right? And so we look for this pattern. This is the pattern. So chimpanzee is different from Neanderthal, and then Neanderthal is the same with one modern human, but different from another modern human. And this is one of two patterns we can find. This is what we call the Babba pattern. And I'm not joking. And the other one is the ABBA pattern. And so this is where the B persists here and the A persists here. All right? And so you see here that B Neanderthal matches. Think back to British English and Canadian English versus American English. And then the other case is when this is A and this is B, this would be British English and American English sharing something. I don't know what it is. I believe that we're two countries separated by a language, so I don't know if that analogy works. But theoretically, if this is the real history, then you should get as many Babbas as you get Abbas, right? Because if it's a really coin flip, you should 50% of the time the Neanderthal should share with this modern human population, and 50% of the time it should share with that population. They should be the same. All right. Now, when we deviate from, if we take Babba and we just literally count, we just go through your genome and we say, okay, which ones meet these criteria, where there's chimpanzees different from Neanderthal, Neanderthal matches one modern human, another modern human matches chimpanzee, we put those to a side, and then we literally count this pattern with the other pattern. And if the sum is 0, when we compare the two, we take this minus that, then no mating between this population and that population, or this population and one of these populations. Now, if we get more Babba than we do Abba, that means that there's probably gene flow from this guy over here, right? Because we're getting more things that are similar between Neanderthal and this other population than we'd expect. If Babba is bigger than Abba, that means there must be genes flowing between these two. All right? And that's the fundamental insight that was used to identify what proportion of a genome is Neanderthal. All right? And what you do is you take chimpanzee, Neanderthal, and you take an African population here, and when you took a European, you got more Babba than Abba. All right? Consistently larger, more of these patterns than you get of the reverse. So there's more Neanderthal-like alleles in Europeans than we see in Africans. When you put Chinese over here in the initial paper, you got the same thing. But if you put another African in here, Babba and Abba were the same, meaning that there's no evidence of gene flow. There's not a thumb pressing on the scale of this pattern. If you put a European, and in the original case, they're using canonical genomes, the Chinese genome, Babba and Abba was, again, 0, which means that there's probably the same amount of gene flow occurring into both. All right, so the thumb is on the scale for both of them. All right, and so this is fundamentally how they figured out what proportion of your genome is a Neanderthal or archaic. You can do this with any population. You could put Denisovans here. We could put African here, and we can look at different modern European or South Asian and try to figure out if there's been recent gene flow from Africa. This is generalizable. They do this now with Darwin's finches, and you can find similar papers using this method. It's this simple. It's Babba, Abba, Babba. All right. Now, like I said, if there's no interbreeding, these should be the same. So what do we find when we look at these big panels of human genomes? Is that we see the relationship, the Babba relationship with with Yoruban populations sort of stand-in for African populations at this period in time, is roughly 650,000. And that's, when we look at ABBA, it's the same. So this would be 0 right here, if you take ABBA and Babba. What we see is we see an enrichment of European ancestry, of Babba sequence, the Babba scenario, all right? And this suggests that there is an enrichment of Neanderthal alleles found in Europeans versus Africans or other populations. So in fact, what we see is we see evidence of this introgression, okay? This is actually important because it looks at a statistical level without identifying any single variant, all right? Because most of your genome is actually identical to Neanderthals, in the same way that most of our genome is actually identical to chimpanzees. Right? Most of our genome is really conserved. And so to get a full-on proportion of your genome, you need to think about these statistical things. Right? So this is a very statistical argument based upon doing these counts. All right? Question?
Well, it shows my ignorance. Is it possible that the European shows up more because that's where the Neanderthals died out and it's just more recent than in the other populations?
So the question is there a geographic component to this? Is the reason why Europeans show more, seem to share more patterns of recent ancestry, the Neanderthals at loci throughout the genome than Africans is because Neanderthals were more recently found in Europe? Partly that's true. That's partly, there's a spatial component to the idea that European Neanderthals were found in a restricted area in Eurasia, and modern human populations would have had to be there to interbreed with them. That isn't to say that Africans probably don't have any archaic ancestry. They probably have archaic ancestry. It's just not Neanderthal. It's probably some other population that was in Africa, right? Because there's a restriction of geography and opportunity. Right? You have to be at the same place at the same time. And so I'll talk about this a little bit at the end. All right? And so this is one way in which we find the percentage. So this is when 23andMe says you have a percentage and you're 4% or less than 4%, or on average Europeans are 2.8%. These values fluctuate dramatically, actually, if you read the papers. It's from doing these sorts of analysis, this incomplete lineage sorting analysis. Now, most people are interested in individual variants. This allele comes from Neanderthal, and this one does not. All right? And so when you go into 23andMe, and again, I'll use myself if you guys don't mind. I'm a little self-centered, I guess, that way. We can, you guys can ask me questions about your stuff or more questions about just me as an example. You go into 23andMe, you get this little thing that says you're less than 4% or you're less than 3% Neanderthal, and then you have a lot about variants. Right? You have X number of variants, or you have this variants and those variants. Those are the mutations that you share with Neanderthals. Okay? So for me, I have 280 variants that I share with Neanderthals. Remember how 23andMe does the research into your ancestry is that they look at your genome and they look for, when I originally did it, it was 600 and some thousand different positions where they look for positions that I might have one allele and somebody else has a different allele. And it's important for differentiating between where we come from geographically. They now have a chip that does, I think, 1.6 million or so. Some of these are unique and found only in Neanderthals, all right? And the modern humans in which carry these alleles. So they're markers. It's like finding what we call a private allele that's restricted to a certain geographic area. If you carry that, you have ancestry from that area, okay? So they look at these in Neanderthals, and apparently I'm in the 53rd percentile of 23andMe customers. One thing we have to note is that the majority of 23andMe customers have European descent, so I'm probably much higher in terms of my Neanderthalness amongst the global population, because you have about 1 billion people in Africa who probably don't have Neanderthal descent, or much of it, and so I immediately jump above them in terms of how much like a caveman I am. Not necessarily something to be excited about, but that's the case. So we have to remember that this is all about our reference to customers, okay? And that this estimate comes from this incomplete lineage sorting. So why are we interested in variance? Well, one thing is that it allows us to compare ourselves to our kin. I am the second to least Neanderthal amongst my family. Papa Kitchen here is slightly less Neanderthal than me, but I definitely remind my mom and my sister that they're significantly more Neanderthal than me. All right, what does this mean? I don't know. I do take comfort in the fact that my colleagues, Andrew Forbes and Cornelia Lang in biology and physics, are both more Neanderthal than me, reconfirming that what I always suspect is that as anthropologists, we're very cultured. Now, again, just kind of want to reiterate that, again, these are just the variable parts. The things that are definitely Neanderthal, that are carried by Neanderthal. Most of my DNA that's Neanderthal is identical to what I would have inherited from my modern human ancestors. So it's really hard to figure out what's actually Neanderthal and what's not. And that's why you need that incomplete lineage sorting to get a statistical approach to look at this. All right, and so we'll talk again about one trait that we really thought might give us insight. One gene might give us insight, and it turns out that every human we've ever sequenced that doesn't have a pathology, including Neanderthals, has the exact same DNA sequence there. And so it tells us nothing, because we need variation to actually figure something out. All right, so why do we care about variance if we had these statistical estimates of 2% of 3 billion nucleotides come from a Neanderthal? Well, it's because we want to know what these variants are, what they do, right? What kind of phenotype, what kind of phenotypes do we have? It turns out that, for example, Neanderthals have a variant that they pass into modern humans. That means we have less back hair, all right, amongst men. I do not have this variant. Luckily, I apparently have other variants that make my back hair very, very mild. But we have all these other, phenotypes that Neanderthals have, the Neanderthal alleles have affected. All right, so there's an allele that if you have the Neanderthal allele, you gain 0.1 inches in height on average. And then there's another place in your genome where if you have a Neanderthal allele, you're 0.1 inches shorter on average. And so you can see oftentimes these things come out in the wash. All right, but what were really interesting, and these are all kind of cute and interesting. Do you sneeze when you eat dark chocolate and things like that? But do some of these Neanderthal genes actually help us? All right. Do individuals with Neanderthal genes, do they have an advantage in certain situations, in certain environments? And that's really kind of the million-dollar question that people are looking at. Okay. All right. My animation failed me. That was supposed to come up last. All right. Well, it turns out that in some populations, the Neanderthal alleles do provide advantages. In this study, this was the Denisovan, a comparison between Tibetan population and the Denisovan genome with local Han Chinese population in the Denisovan genome. They found in one gene an allele that was incredibly divergent from anything you see in modern humans, but matched identically what was in the Denisovan. And that was a gene that was under positive selection for adaptation to high altitude. So one of the reasons why almost all, this large population of Tibetans, they almost all had this allele, or a very closely related allele, that they definitely inherited from Denisovans. So part of their adaptation to high altitude was they interbred with Denisovans, and then those individuals that had this Denisovan allele were more successful at living in high altitude. And so this allele persisted in the population and spread, and now almost everyone has it. All right, so this is one example of this. All right, we now know of multiple examples where we think this is the case. In some populations, we have examples of things that might actually lead to some disease. So Mayan populations have some archaic parts of their genome that we think are related to an incidence of diabetes, for example. So that might be a mismatch between the current environment and the archaic component of their genomes, right? Now, the important thing about this is to remember is that these populations persisted in these environments for tens of, if not hundreds of thousands of years. So they would have been very well adapted to those environments. Modern humans encountering archaic human populations would have been new on the ground, maybe couple thousand years in that environment, not enough time to actually adapt. So interbreeding with existing archaic populations that have adapted genetically to their environment might be a way for those expanded modern human populations to capture some genes that give them an adaptive, adaptive fitness to that environment. All right? So instead of having to wait hundreds of thousands of years for these alleles to pop up and then increase in frequency in a population, you could just grab them by interbreeding immediately. All right? And so this is what we think. Question?
Has research shown anything yet about the nature of the adaptation? Taking hemoglobin or anything like that? Is that all?
It actually thins the blood, which is interesting. So it actually doesn't increase hemoglobin. It actually thins the blood, and that apparently has some other effect on the metabolism. I've forgotten most of the biochemistry I ever learned, and so.
So we care less?
No, it changes the viscosity of the blood, and then this affects the gradients over where O2 is coming in, and it's much more complex than it's in a pathway that all these things work out. But it's kind of what was interesting was that it was a simple thought about how it would work. Actually, it works the opposite of that, and that's kind of what threw him for a loop. We do think that there's a enrichment of archaic alleles in parts of the genome that are related to the immune system, to the immune system. So we think that this might be related to the fact that these Neanderthal populations were very, very well adapted to local disease environments, and that when modern humans came in, and if they interbred, then those were the parts of the genome that were positively selected for, and so we quickly just incorporated those alleles to our genomes. The strongest signal of selection in the human population globally that we see everywhere, it's a different signal everywhere, but it's always there, and it's a different part of the environment, is to our local infectious disease. So every population on Earth that's been in place for several thousand years has shown adaptation to local infectious disease.
To which disease?
To local infectious disease, so worms, parasites, single-cell parasites, bacteria, viruses, those sorts of things. Every population shows it. All right, so that's not surprising that these archaic populations would also have adapted to it, and that human populations, modern human populations, if they interbred, those alleles would be kept around by positive selection. All right, and so this is actually one of the big things is what's the phenotypic effect of all these variants? Yeah, we have them. Most of the genome we get from Neanderthals is identical to modern humans, so it has no effect essentially on our phenotype, we think. But those things that are different, like what's happening? Why what might they be causing? One thing that we still have a hard time figuring this out, because there's this speech gene that you guys might have heard of called FOXP2. It's involved in the fine motor control of producing speech. We're certain that it was a speech gene. There's modern human families that have mutations in it that are unable to produce speech or comprehend speech. So we looked at chimpanzees, and they have a different gene sequence for it, and so we thought that maybe this is the case. When we looked at Neanderthals, exactly identical to modern humans down to the single nucleotide. So this won't tell us how Neanderthals are different. Could Neanderthals have spoken? Genetically, it looks like, at least from this gene, it does. But there's still a lot of outstanding questions that even with having an archaic genome and even having human, essentially, many of us here are human Neanderthal hybrids. Like, we still haven't figured out what the actual variants are doing and what are the things that make us anatomically modern human versus the things that made Neanderthals Neanderthals. All right, so this is like the million-dollar question. All right. So one of the really cool things to think about when we're looking at these variants and we're looking at the genome and where in our genome, so this is a map of all 23 chromosomes. This is me comparing with my mother, since she doesn't have a Y chromosome. We don't have that comparison here. Is to think about the size of the blocks of inheritance that we have in our genomes. All right? This tells us something about how recent this occurred. So I inherited all this from my mom. You'll notice every single chromosome is a full block. Right? It's 100% inheritance from my mom. I have the exact same set from my dad. It's the complementary set. And over time, however, as we go through multiple inheritances, these blocks get divided up via recombination. All right, so this is me in comparison to my sister. So you have to go up me to my mom and then down to my sister. You'll see that on average, we still share 50%. But it's not equal. So these are places where me and my sister have entirely different DNA sequences. And these dark purple places are places where we inherited the same alleles from both of our parents. All right, and so over time, as my niece and nephew, we'd share half of what I share with my sister. And there'd be a lot more spaces here where we share none at all. And so these inheritance units get chopped up every generation. Okay. Now, what does this mean for looking at our archaic ancestry? Is that the size of these inheritance blocks tell us something about when this occurred. All right, so the longer back in time, the smaller the inheritance units we're going to see little, if it's really ancient, we're going to see little bits of Neanderthal DNA surrounded by modern human DNA, and then just little bits here and there. If it's more recent, like if it was last generation, I'd have full chromosomes that are Neanderthal. All right, so we can figure out the timing of these events by looking at the size of the blocks. Another thing that affects the blocks is that if we have a block that has variants that are adaptive, those will sweep through the population and they'll stay large. All right? And we can use that then as a signature of figuring out what's under positive selection. Okay? And so this is yet another way that we can look at the Neanderthal genome, is by looking at these blocks of inheritance. In addition to the number of them, we look at the size of them. Okay? And they can tell us a lot of information about what's going on. Now, if we look at my genome, again, because my favorite subject is me, we can see each one of these little blue lines is a location where there's a variant that I inherited from a Neanderthal. And the first thing to note is that they always automatically put it on the top allele. So it's not like I inherited all of these from my mom and then a few from my dad. If the top chromosome is the one from my mom, then the bottom one's from my dad. That's not the way it works. They just map them all the variants to the top. And then occasionally you see double variants, like right here or right here. These are places in my genome where I have Neanderthal from both parents. All right, so the thing to remember is that I don't have a lot of clusters of blocks of variants. They aren't always clustered together. One second. And what that means is that my Neanderthal inheritance comes from a long ways in the past, tens of thousands of years. Question?
Is there a reason why the X and Y chromosomes do not have any variant?
Yeah, it seems like there might be some. So the question is why don't X and Y have any evidence of Neanderthal introgression? Well, the parts of the Y chromosome that have been sequenced for this ancestry are non-recombining, so there's just one big inheritance block. All right. And so this would have had to we would have had to have a Neanderthal ancestor that's unbroken in my male line. So my dad's 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 dad had to be a Neanderthal. And we don't see that. We have no evidence of the Neanderthal Y chromosome. The same way we have no evidence of a Neanderthal mitochondrial DNA, because it had to be moms. Another thing is that we're not sure, but it's possible that there is sex-biased introgression. All right, that there are genetic incompatibilities that can occur between the sex chromosomes that favor matings by, for example, Neanderthal men and modern human females. If that's the case, you would not see any introgression of X chromosome into modern human populations. Alternatively, you could have the mating, but if the X chromosome of Neanderthal is incompatible with the X chromosome of the modern human, you might not actually, that individual might be not fertile. So you would never actually pass on the X chromosome. So there are these barriers, and these barriers to reproduction often occur in the sex chromosomes. All right, so we think that there might be a partial barrier. This might be evidence of a partial barrier, so that not every single crossing of a modern human and Neanderthal always produced fertile offspring. Or if they did, those offspring weren't as fertile, as fit, and different formations of pairings, or between an anatomically modern human and a Neanderthal. So that's a good question. All right. So the thing though is that these are all sprinkled throughout, not through the sex chromosomes, but you get this sprinkling of Neanderthal throughout. It's another thing to note that the places where I'm Neanderthal are different than the places where my sister's Neanderthal. This is because each one of these loci has a different, even though we're closely related, they have different histories. They're moving through, some of these I got from my mom, some of them I got from my dad. I'm confined to only getting what my mom and dad have, but I get a subset of them. And they get mixed and matched. And I get some from my dad, and what I get from my dad is different than what my sister got from my mom. So we can never have more than they had, but we're going to have different subsets of it. And what this means is that scientists have gone back and they've reconstructed Neanderthal genomes from just modern humans. They've gone and they've taken this little bit from you, and 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 they've put it together, and they got about 50% of the Neanderthal genome. It's because they know what the Neanderthal genome looks like, and they're able to reconstruct about 50% of it. So if you know, at some point in time, if we ever wanted to reconstruct a Neanderthal, and for whatever reason the servers with the Neanderthal genome went down, we could get pretty close. All right, and that's because this Neanderthal introgression, these genes have spread throughout, and they all have their own histories, okay? And this is actually really interesting because it suggests that even though we don't have genomes of other archaic humans, we might actually be able to reconstruct them from what we carry around with us today, which is pretty cool. All right? And so to kind of summarize, to finish up, we've now taken this from looking at Neanderthals and Denisovans to looking at other populations from which we have ancient genomes, the most prominent of which is Europeans. All right, so you guys might have seen a slide like this before, where using reference genomes and genotyping kits like 23andMe from populations where we're pretty sure the people have lived there for generations, like great-grandparents have lived there in Europe, we can create these genetic maps from these reference data sets. All right, and you can see here, like, these are Spanish, and they're close to Portuguese, and then France, individuals from France are up here, individuals from Great Britain, and whatnot. And their genotypes, and this is what we call a principal component analysis. So you take this huge dimensional data, and you figure out what's the simplest two-dimensional way of catching all the variance, all the variability in that data. And first and second principal components are latitude and longitude in Europe. So we can reconstruct the map of Europe by getting genotype data from individuals whose grandparents had lived in the same country they do now. All right? And this is essentially how 23andMe goes about painting my genome as coming from certain parts of the world. All right, they have this huge reference genome of people who say they came from where, and you look at my genome and you say, okay, where do these variants, who do they match with? And then you paint my genome so you can see that all this blue is European, and then I have a little bit of West African here, which is possibly from my dad's side, or it could just be a bad reference set. I'm not sure. But this is exactly the same. This is essentially what we do today. We could do the same thing if we have archaic European populations. All right? So what we could do is we could go and we could sequence some ancient Europeans and create a genetic map and say, like, okay, so ancient Europeans that are hunter-gatherers come up over here. If you're a early farmer, you come up down here, and then you throw me onto it and see where I come up and look at my variants and map them. You can then paint my genome the same way. And that is exactly what we do. All right? So this plot, which is a poor version of the previous plot of Europe, includes some ancient populations. So it includes individuals from 8,000 to 9,000 years ago. And they look very distinct from modern Europeans. And using this map, you have some ancient northern Eurasians here. You have some Scandinavian hunter-gatherers, and you have Western European hunter-gatherers, and then you have early farmers coming from the Middle East. And this is kind of like a Mediterranean sort of Middle Eastern gradient here. Trust me, if you don't see anything, it takes years to be able to read these. Essentially, this relates to a scenario where you have hunter-gatherers in Europe, and then you have some early farmers moving into Europe from the Middle East. All right, so the hunter-gatherers are distantly related from the Middle Eastern populations. And you have genetics from the Middle East coming into early farmers. These farmers then spread throughout Europe, and this is the classic story of how Europe was populated. And then you have these northern Eurasians moving in over here. All right, And we can look at individuals' genomes and we can match. We can paint your genomes by variants that are at high frequency in this population, at high frequency in this population, at high frequency in this population. And we can say what proportion of a European individual's genome probably came from each one of these migrations. All right? And there's a popular website called Family Tree DNA that you can import 23andMe data to it, and they'll do this. All right? They'll map your genome, look at your variants, and figure out what proportion of each of these migrations are you. And as completely predicted by the fact that I grew up in Indiana and I now live in Iowa, I'm a farmer and a hunter, and I'm not much of an invader. I haven't moved very far. So you can do this, and this is just taking the idea of an archaic population. Since we're not dealing with species, we're dealing with populations. Neanderthals aren't in our species, they're just a different population. And moving it to this idea that, looking at populations in time. So different populations that are diversified. So hunter-gatherer Europeans, you could do the same thing in Africa, you could do the same thing in Asia, as long as we have access to these ancient genomes. All right? We're doing the same thing in North America. I'm trying to do some of that in Canada. All right, which takes me to one of my last little pet toys when thinking about archaic ancestry is lice. I would imagine most people who have children have thought about lice a little bit, or at least interacted with lice. Lice are really fascinating because we've had lice for a long, long time. I'm sympathetically scratching my head. And in fact, we had lice before we had clothing, and we can date the origins of clothing by looking at when clothing lice show up. But head lice are fascinating because head lice show an amazing amount of diversity. There are multiple lineages of head lice that have common ancestry only hundreds of thousands, if not millions of years in the past. All right? And if you took a louse from a human, and a louse from a chimp, and a louse from a gorilla, and a louse from an orangutan, and you looked at them and compared their relationships to each other, the relationships between these lice would be identical to the relationships between these apes, suggesting that all of us have had lice for millions of years, and our lice are evolved to us. Now, these are one of these take-home facts that you can gross out people. There's been one host jump amongst lice. Anyone know this or guess? The human pubic louse is a gorilla louse that jumped from gorillas to humans in the last 2 million years, probably about 2 million years ago. It's probably not what you think. It's probably because humans and gorillas were either in conflict at the time, or gorillas make these really amazing nests on the ground, and humans at the time would have been much more ape-like, and we might have actually slept in one of their nice beds that they made. All right? Now, so that's the only example of species jumping hosts. Now, one question we have is that we don't know how long humans and Neanderthals interacted. We know that they interbred, and we know that humans and archaic. Denisovans interbred. We don't know how long they would have been in contact. It might have just been, mate and go. However, we do know that for you to get lice, you should, there is some contact, but it's repeated contact is typically necessary. We think that these deep lineages of lice that go back millions of years are unlikely to have been carried in modern humans alone. Our simulations suggest that lice on modern humans, other lineages would have been lost. In the same way that when we think about incomplete lineage sorting, we get the loss of one of those alleles, we'd expect the exact same thing to happen to lice. So why are there 3 types of lice, of head lice, lineages of head lice in modern humans? We think it's because Neanderthals and some other, in this drawing we call it Homo erectus, some other archaic human carried these lice, and they became the only lice on each of these. And then when modern humans encountered them, and possibly interbred with them, or were in conflict with them, they jumped over. All right? And so we think it's not just genes that jumped over, we think that it might be other things like parasites as well, in this case lice. All right? And so this is one of the things that we can use then to figure out when these occurred, when these jumps occurred and where. So each one of these arrows is a possible jump. The more modern genomes we get, the more evidence we're finding, and the more fine scale our evidence can be because we're characterizing more and more populations as opposed to just being like all Asians are Han Chinese and all Europeans are Mormons in Utah, which is what the first studies assumed. All right? we end up finding more and more of these introgression events. And in fact, we're finding introgression events probably in African populations. We have very, very old alleles in African populations that are older than we'd ever expect. It's just that we don't have a comparative genome from an archaic African to compare them to. So they just look like very old pieces of DNA that are way outside of distribution. So this suggests that there was probably archaic admixture in Africa as well. So this isn't just a non-African story. This is all humans. Wherever they encountered archaic humans, probably interbred. That's a very strong statement, but I'll say it here. It's not in print, so don't hold me to it. So in conclusion, it's most human populations probably have some archaic alleles, right? Humans probably interbred with other humans whenever they found them. It's unclear what the phenotypic effect of these alleles are. We have no idea, like if they actually make you more adapted or less adapted to your environment. We'll find out more. We'll figure out more of this as we get more ancient genomes. Each one of our relationships to our archaic ancestors is different and unique. So we all have different ancestry at different locations unless you have an identical twin or a clone. And ultimately, we're all a little archaic, all right? And Neanderthals are still alive. They're just us, all right? And so this is the case for all the archaic human populations. So if you have any questions, I know I went over a little bit. Please let me know.